Welcome back everyone to my third video in our series of short science talks about epigenetics. As we've started to think more about our lives in terms of epigenetics, it becomes clear how much we really do determine how healthy we are. In this video, we're going to explore a powerful twin study, and we're going to look at how our cells choose which genes to use. As we discussed in my first talk, identical twins have exactly the same DNA, but twins get much less similar as they get older, as they express their DNA more and more differently. There are many visible differences in identical twins as they age, but can we quantify this scientifically? We're going to address this question and much more in the next 5 minutes. My name is Robert Needleman. I'm a molecular biologist and biochemist. Welcome to Empower My DNA. Here's a powerful twin study that was published. Epigenetic differences arise during the lifetime of monozygotic or identical twins. In the study, they looked at many pairs of identical twins and they looked at their DNA. They compared several pairs of the chromosomes of the twins. Yellow shows where epigenetic tags are in the same place for both twins. So 3-year-old twins are using their DNA quite similarly. In contrast, red and green shows where epigenetic tags are in different places. You can see from these pictures how differently 50-year-old twins express their DNA. Here's chromosome 3 pairs of the same twins, so you can see them side by side. Again, yellow shows where the epigenetic tags are in the same place for both twins, and red and green show where epigenetic tags are in different places. It is very clear from this powerful study how differently identical twins use their DNA as they get older. Here's a perfect example of 2 identical twin women who look very different as they're getting older. Now, the science that's coming next is really going to blow your mind. Here's another amazing example of identical twins, our organs. We have exactly the same DNA in our heart, in our eyes, our kidneys, our lungs. Although our organs look very different and perform very different functions, they have exactly the same DNA, but they use their DNA differently. This brings up an obvious next question. How do our cells decide which genes to use? Now, to answer this question, we're going to look at a publication from a brilliant scientist many of you probably have heard of, Dr. Bruce Lipton. Published this paper in 1977, a fine structural analysis of normal and modulated cells in myogenic cultures. Dr. Lipton used stem cells, which are genetically identical clones. They all come from one cell. He split the genetically identical cells into 3 culture dishes, but each dish had a different environment, A, B, and C. So we have the same cells in 3 different environmental conditions. To me, the results are profound. In environment A, the stem cells form muscle. In environment B, the stem cells form bone. And in environment C, the stem cells form fat. So what controls the fate of the cell? The environment. This brings us to an important publication from Dr. Nihut, Metaphors and the Role of Genes in Development. In the paper, he makes the potent statement, when a gene product is needed, a signal from its environment, not an emergent property of the gene itself, activates expression of that gene. In other words, genes cannot turn themselves on and off. Genes do not self-activate. Genes are selected in response to the cell's perception of its environment. This is a profound piece of information on our journey to understanding how cells choose which genes to use. Our DNA stores information in the form of genes and in other ways, but the DNA does not choose its genes. This is very empowering for us because we can influence which genes we use. We can say things like, my cells choose the best genes for my health and longevity. In my next videos, we will start looking at how our cells actually read their DNA and look at many examples in nature of epigenetics at work. We will continue to focus on our overall goal, becoming more and more empowered in our own lives. Thank you very, very much for joining me. Have a most incredible day.